I'm really honored to be invited to this edition of Montpellier Omics Days. And I really want to congratulate all the students who have participated in the organization of this event. You really did a great job, impressive job, bravo. I'm really glad that you gave me this particular moment of the day because since this morning, we have seen a very beautiful spectrum of what can be done using omics data. And there is something which is really crucial to notice maybe, is that there are some questions that have been asked many times, independent of the context of the research. And that's what I'm trying to talk to you about today. In fact, I'm not going to present you any particular results or a methodology, but I want, in fact, to invite you to a reflective journey around the analysis of the omics data. So we will see together in the next few minutes what are the important aspects when it comes to analyzing data, such omics data, and working and collaborating in an interdisciplinarity or in multidisciplinary studies. I have chosen to use an example, a research question that I've worked on during my PhD candidature, which is B-cell receptors repertoire analysis. I find it quite interesting. I hope that you find it exciting as well. So first of all, we will see together very briefly some basic concepts in immunology in order to understand the research question, and then the main focal point of this presentation would be the questions and the challenges that we're facing while trying to applying the knowledge coming from analyzing omics data into the clinical context. So let's start with some notions in immunology. As you probably may know, the human immunity system has two levels of protection. The first one, which is innate immune response, is actually responsible for protecting us during the first critical hours and days of exposure to a new pathogen. It is not really specific, and it's really important, but it's not really specific. So it cannot remember what has been seen before. On the other hand, we have adaptive immune response, which, as it says by its name, it adapts itself to the pathogen that we encounter in the environment, meaning that it can remember and quickly destroy what has been seen before. Very interesting quality for the immunity system. So, as you might imagine, it's a very complex system, and we definitely won't have the time to see all of the details. And that's why I've chosen to talk to you about one of the stars of the immunity show, which is B lymphocytes or B cells. B cells are one of the major components of the adaptive immune response in the human. And they have some structures at their surface, the receptors, that are actually the structures that recognize and interact with the pathogens. So the receptors, as I mentioned before, at the outer cell surface, have two main components. The immunoglobulin protein, which is actually the recognition unit of the receptor, and the signaling subunit integrated into the cell surface. So immunoglobulin is a very interesting protein. It's a heterodimer. It has two heavy chains and two light chains bind together with disulfur bonds. And the interesting thing about these particular molecules, which are really famous because of their shape, is that they have a variable region which interacts directly with the pathogens, and a constant region which has a more structural role. So you have already two main pieces of information about the adaptive immunity, which is, first of all, it is really, really specific to the pathogen. And also the interaction and recognition it's done by the receptors. So logically, what do we need is to have enough variety of receptors in order to detect the endless pathogens that are in the environment. The question is, how much diversity is enough to be sure that we are safe? The answer is, I really don't know. And this is really hard to evaluate what we actually need. And as you can see in the case of pandemics or like what we're living right now, it is not an easy question to be answered. However, already we have a very amazing possibility, a very amazing variety within our organism, because we can already protect ourselves from a great part of environmental dangers. So the question is where this diversity comes from. Actually, there are some studies that suggest we have something around 10 or 11 unique sequences for immunoglobulin, which is a lot. And we definitely don't have the same amount of genes that code for each specific immunoglobulin. There is a very elegant way in our organism to create such a huge diversity. We will see a part of that today. So, in fact, the immunoglobulin heavy chain, we just look into the heavy chain, is encoded by three, the variable region is encoded by three gene families: the variability, the diversity, and the joining family. So one of the main sources of diversity in this case is selecting randomly one member of each of these families and join them together. In this way, we create a combinatorial diversity. And once we have that, knowing the fact that the junction is not really precise, we have another source of variability in the junction between V, D, and DJ part. So this happens in the earliest stage of B-cell development. And well, once our receptor, or immunoglobulin in the receptor, encounters an antigen, this part creates an stimulation, antigen stimulation, and B cells undergo activation, proliferation, and further differentiation, allowing the secretion of the soluble form of immunoglobulin that I'm sure you know them, antibodies. So at this point, in fact, when B cells are exposed to antigen, naive B cells genes that encode for immunoglobulin undergo multiple rounds of rapid Darwinian mutation and selection, what we call the somatic hypermutations, which are mainly the point mutations, in order to increase progressively the affinity of the receptor for the given antigen. This means that from a precursor cell, we have the development of a cell lineage. And in each step, we try to increase the affinity toward the given antigen. And in this case, we will be able to protect the organism better. So here, what we see is a B-cell lineage. So the collection of one cells and all the descendants during the affinity maturation creates a B-cell lineage. And at a given time, when we look into the immunity system of an individual, we can see a collection of these lineages. And the collection of these lineages creates what we call B-cell repertoire. And why bothering studying B-cell repertoires at all? Well, they have some interesting information, especially for the clinical point of view. In fact, for a healthy individual, we can imagine that you need a great variety of the B cells and B-cell receptors, which means antibodies, because you are facing with different pathogens in the environment. In fact, how we qualify a repertoire when somebody is in good health is that we think that it contains a high diversity and low clonality. I explain it more. Because of some types of situations like age, some kind of disease, or even vaccination, we see that there are some B-cell lineages that develop more than the others, and they take the space, they take the repertoire. And in this case, we decrease the diversity while increasing the clonality, meaning that I have a clone or a B-cell lineage, which is expanding. So if we want to illustrate that, we can see that a highly variable, diverse repertoire, which is the case of a healthy individual, is qualified as a polyclonal repertoire. While when we have some lymphoid malignancies like leukemia, we see something which is more on the other side of the spectrum, very low in diversity and very high in clonality. So these information provide a valuable indicator of the human health, and also it can be really, really helpful for diagnosis, prognosis, and also the follow-up of the patients after a given treatment. So it is important to study B-cell repertoire. That's the conclusion. So omics data have given us the opportunity to discover and explore the repertoire that we couldn't have imagined before. And it's great, it's a good news. It also means that we need some tools for the data exploration and to be sure that we are gaining some insight from studying these data. And when we talk about developmental problems and omics tools and application in the clinical context, we're definitely talking about interdisciplinary and multidisciplinary studies. That's the first point that I wanted to talk to you about, because you will definitely, as young scholars, work on these environments in general. And it's really good to have an idea about the challenges in these kind of studies. So let's think about a question that we want to answer in the clinical context. And we need to develop a tool a software, a computational tool for that? What are the requirements for a tool to be used in the clinical context for the clinical purposes? There are so many. I've chosen some of the important ones. So one of them is that we have to be sure that what we see as the results are reliable. So no need to go further. This is obvious for everyone. It should be really reliable. On the other hand, we have another problem, which is we don't have so much time in the clinical context. If you remember, today, Benjamin has showed us the differences between two algorithms, two different softwares. One of them do the same task in 1 hour, the other one do the same task in few seconds. In some cases, it's really interesting. We say, okay, it's better to do the things in a lower amount of time. But sometimes it becomes so important that even our tool is not usable in time, which is allowed in the clinical context. We can't definitely use it at all. So it's not just a luxury to have a very fast and accurate tool, but it's a necessity in this context. Another thing which is so important, and sometimes we tend to neglect it, me myself did this before, is that we develop some tools. They're marvelous, they work great, but they are not user-friendly. They are not designed to being used in the clinical context, meaning that sometimes we need a very, very high computational resources, which is not always available. And to be honest, it's rarely available in the clinical context. Or we need very high computational skills or informatics skills that people in the clinical context really don't need to have them. Their job is something else. So mainly, these are reasons why it's not really obvious to transfer what we have in the research environment to the clinical and more practical environments. So there are endless amounts of challenges, but what we're going to talk about today, I've tried to select 2 of them, which are really common. The first one is the ambiguity. And this morning again, Lucy she was talking about the fact that there are some concepts that don't have the same definition among different experts. And it seems a little bit odd, but that's a very common issue. In fact, in our context, in our example, is the clonality, but we have to be able to give a very precise definition for each of them, which is not always obvious. Because, for example, for instance, we will take the example of the clone. So we say that a clone will represent a B-cell lineage. Okay, what we define as a lineage is a cellular concept. What do we have is a proxy of the cellular concept, which are the sequences. And how you will define the clone that represents the B-cell lineage using sequences that it reflects in the best way the biological concept. So many different interpretations, many different debates. It's not an easy task to define fundamental objects when you're working in these kind of environments. So that's one of the potential issues that you may face one day. The other one is the complexity. of these kind of analysis. So imagine that you have a very clear research question and you want to answer that by developing a new tool, which is amazing, work really fast, and it is great. So first step is that you have one idea, you go through the development stage, and in general, when you want to publish your results, you do some verifications. In general, for example, you have some data that have been already analyzed and you compare the outcome of your method with the already published ones using these data. And you go through a loop of analytical validation and verifications. And once you establish a good, a solid base, in general, you will transfer that to into the clinical context in order to receive a clinical validation, meaning that is my analysis valuable in this context? Does it give any further insight about what has been done before, about what we're searching for, that's new, that's helpful? So in this part, you have to be with the users and have a very tight flow of interaction. So they give you their feedbacks, you work again on your pipeline, and actually it's a never-ending loop. The problem is that sometimes the communication between these two parts, it becomes so hard that this loop stops. And that's how in real life, many collaborations stops. There's no way to continue collaboration when different partners cannot really understand each other and understand each other's need and requests. And well, that's a little bit sad. So let's take an example and see, okay, we want to answer this question. What are the steps? What in the real life, what we are going to do? So the question is, you know already what is a B-cell receptor repertoire. You know why it is important. The question is, how can BCR repertoire organization and intra-clonal properties be better explored to produce helpful tools for the medical community? An interesting question. So what would be a pipeline for that? What would be different steps? So in general, what you have to see what is available as the input data. In my case, it was the blood samples. So you extract the sequences that you're looking for, the heavy chain, because it was, in my case, the heavy chain genes of the immunoglobulin. And the next step is try to understand these sequences. Traditionally, the first step is to try to annotate these sequences and determining, okay, in this sequence, what is the V gene, what is the D gene, what is the J gene? And this is a very important part because it's the starting point of creating the lineage that we have talked about. I'd like to open a little side here, saying that for this task and many others in immunogenetics, IMGT, the International Immunogenetics Information System, is the international leader, in fact, in the domain. It has been created in 1989 by Marie-Paul Lefranc in Montpellier. And if you are interested in immunogenetics and all the analyses related to immune responses from genomes to proteomes, I really encourage you to follow their inspirational work. And honestly, it was a dream for me to work here, and I'm really glad that finally I can do it. So we have our sequences annotated. Now the question is, how can I select and collect all the clonally related sequences and find what are my clones, what are the infrastructure of a clone. Once I have that, I will carry on the clonal analyses and the intra-clonal analysis. That's the very simple way to evaluate if these kind of studies would be helpful to be used in the clinical context. So one possible solution for this specific question would be visualization. Why? Because I have a very clear question. I know what are the steps, and I know that I have to maintain a collaboration between people with very, very different expertise. Different expertise means different vocabulary. When you're talking about the same concepts from different perspectives, we don't actually mean the same thing. And sometimes it's really problematic. So what I've personally found really helpful in this case was visualization, meaning that we try to translate all the concepts that we're trying to work on, all the objects of the study, into some visual representation. In this case, we will be at least try to make sure that we are talking about the same thing. So during my PhD, I've participated in development of a tool which is called VCLOD, that in fact tries to do these different steps in a visual manner in order to be used in the clinical context. So here we will briefly see what are the different modules and analysis proposed by VCLOD. I don't want you to, go through details, but just thinking about the possibilities that we have to communicate between computational experts and medical experts. So here, what we are looking at is a repertoire, and very bad news. This is coming from a patient who has a very severe leukemia. In fact, what we are looking at, the yellow circle is showing the expansion of one clone, which somehow represents a tumor. And you can see that it's really, really huge. And you don't really see the variability that you want to see in the case of a healthy individual. So here we provide a quick visual indicator of the diversity within a repertoire, and the clinical expert can go through different details about each sequence and about the distribution of different clones. The thing which is important is that this tool can help the clinical expert to go through more details and look into each clone and see different subpopulations of the clone. And if you remember, when we talked about affinity maturation and development of the receptors that are more specific, more finally specific, we created a tree. So we thought that, okay, maybe it would be interesting to look into the tree and try to work on it. At this stage, we don't really know what are the interesting aspects of this tree to be explored. So what we let the medical doctors and biologists, what we let them do is to work on their data using these structures and giving us the feedback, saying, okay, if they see any patterns, for example, in the tree topology, we can go even further and see more details about each tree and the evolutionary history of the tree, and doing even more refined analysis. So this is an ongoing project, meaning that we try to look into different types of data, different types of pathologies, in order to see if we can create some meaning for our data. So there are different types of visualization, and it is always interesting to add more of them. That's what we're trying to do. And that's what, in fact, many different labs in the world try to carry out. And what we try to do is to translate it, the very complicated, hard to manipulate algorithm, into something which is user-friendly and has a chance to be explored in the clinical context. So the only thing that I wanted to tell you, for you, because you are young scholars, is that omics data has created many opportunities and also the challenges. If there is something to work on, and if there's one thing to work on and be sure that you will someday need it, is your communicational skills, because it's not only auxiliary to your work, it becomes part of your work, because the subjects that are working on are so complex that a single expertise, a single field, a single domain wouldn't be able to answer that. We need different expertise, we need different domains, we need interdisciplinarity and multidisciplinarity studies. So the communication is the key. And here I want to just thank the team behind B-cell, the example of the presentation today. And I wanted to tell you that it was a very tough job to work during years together and to establish a common language. And I hope that it was at least a little bit helpful for you in your future career. Thank you.